Welcome to This Explainer. Today, we're uncovering the fascinating science behind some of the most talked about, wildly successful drugs in the world right now. We are decoding the Goldilocks molecule, which is better known in the scientific community as therapeutic peptides. Let's jump right in. So, I want you to ask yourself a slightly eccentric riddle. What do a venomous lizard, the 1920s, and a $70 billion market all have in common? It sounds like a bizarre setup to a joke, but the answer actually lies in a specific class of molecules that have quietly, almost invisibly, revolutionized modern medicine over the last century. And yeah, you heard that right. $70 billion. That staggering number represents the worldwide sales of peptide drugs in the year 2019 alone. And honestly, that market is only growing, largely driven by absolute blockbuster names you've probably seen in the news or on commercials. Drugs like Trulicity, Victoza, and Ozempic. But what exactly are these drugs? And why are they suddenly absolutely everywhere? To understand that, let's look at the foundational definition. Therapeutic peptides are basically pharmaceutical agents made up of a series of well-ordered amino acids, usually weighing in somewhere between 500 and 5,000 daltons. In plain English, they're incredibly tiny. We're talking much smaller than a full protein, but a bit larger than your standard run-of-the-mill chemical pill. At their core, these are highly precise, microscopic sequences that our bodies already use to communicate, so they aren't some completely alien synthetic chemicals. They're actually the fundamental building blocks of life, just carefully engineered and optimized to help heal us. Here is our roadmap for today's explainer. First, meet the therapeutic peptides. Second, the Goldilocks of medicine. Third, venom to blockbuster drugs. Fourth, hacking the peptide backbone. And finally, the future of peptides. Let's start with part one, meet the therapeutic peptides, the building blocks of life functioning as medicine. When these molecules enter your body, what are they actually doing? Well, peptides commonly act as hormones, growth factors, or neurotransmitters. They essentially travel around and bind to the surface of your cells to trigger very specific actions inside. You can think of them as nature's messengers. They carry these highly specific instructions, acting like specialized keys that only fit perfectly into very specific locks on your cell surfaces. And by turning those locks, they dictate everything from regulating your blood sugar to sending pain signals to your brain. Moving right along to section 2, the Goldilocks of medicine. We're looking for that just right molecule. To really grasp why this is a $70 billion industry, you have to understand the massive problem these molecules solve for pharmacologists. And the Goldilocks analogy absolutely hits the nail on the head here. Peptides sit right in the sweet spot between tiny, generic small molecules and massive, super expensive biologics. Think about it. Small molecules are cheap and they slip right into cells, but they can be blunt instruments, sometimes hitting the wrong targets and causing nasty side effects. Then on the other extreme, you have biologics, like huge antibodies. Highly specific, sure, but incredibly expensive to make, and they can trigger unwanted immune reactions. Peptides sit perfectly in the middle. They have enough surface area to precisely block disease-causing proteins, but they're still small enough to be cost-effective. They are, quite literally, just right. But, there's always a catch. While peptides are wonderfully specific, super effective, and generally very safe because they rarely trigger allergic reactions, they are also incredibly fragile. They suffer from notoriously low stability, a really short half-life, and weak membrane permeability. Nature basically designed them to deliver a rapid message and then instantly dissolve. But if you're trying to make a pharmaceutical drug, that rapid breakdown is a massive hurdle. They just really struggle to survive the harsh environment of your stomach acid or your bloodstream long enough to actually do their job. Which brings us to section 3, venom to blockbuster drugs. Let's trace the evolutionary timeline. It has been a century-long journey to take these incredibly fragile natural molecules and transform them into rugged, life-saving medicines. Back in 1921, Frederick Banting famously isolated insulin from livestock pancreata, which was a monumental breakthrough that saved millions of diabetics. Then, from the 50s through the 90s, scientists got better at identifying and synthesizing more of these natural hormones. But the real game changer happened from the year 2000 to the present. We entered the synthetic era, bringing 33 brand new non-insulin peptide drugs to market. We completely transitioned from just extracting what nature gave us to custom-building complex molecules right in the lab. And to overcome that whole fragility problem we talked about, scientists looked to some pretty unexpected places for inspiration. They scoured the natural world and, incredibly, optimized the blockbuster drug exenatide directly from the venom of the Gila monster lizard. No way, right?
They realized that venom peptides are naturally fortified by evolution to survive extremely harsh biological environments. That makes them incredible blueprints for durable human therapeutics. Oh, and scientists even derived a cutting-edge drug for severe chronic pain from the venom of the magical cone snail. I mean, nature is basically doing the heavy lifting for us.
Let's dive into part 4, hacking the peptide backbone. How do we equip these molecules for survival if we aren't borrowing from venom? This brings us into the chemistry lab. Scientists developed this fascinating, almost coffee filter-like reactor that entirely revolutionized the automated step-by-step construction of peptides. It's called solid-phase peptide synthesis, or SPPS for short. Basically, it allows chemists to build peptide chains exactly one amino acid at a time on a solid resin. Because they do this in a highly controlled lab environment, they can introduce incredibly versatile synthetic building blocks. We're talking entirely unnatural amino acids that biology never even intended to exist. Doing this dramatically extends the lifespan of the resulting drugs. But we're even going way beyond standard chemical mixing now. Using a mind-blowing, genuinely sci-fi technique called genetic code expansion, scientists can literally rewrite the rules of cellular biology. The process is wild. First, they create a non-canonical or unnatural amino acid. Second, they reprogram a genetic signal, something called an amber stop codon. Normally, that codon acts like a red light for a cell's manufacturing machinery, but scientists can actually flip it to a green light, telling the cell to insert our new synthetic ingredient. Finally, they synthesize the new hybrid sequence. They are literally hacking the translation machinery of a living cell to weave tougher, unnatural armor directly into the peptide chain as it's being built.
We are outfitting these molecules with literal molecular armor. Scientists use chemical crosslinks, which is like creating a structural bridge across the peptide, and peptide cyclization, which forces the whole sequence into a closed loop. By stabilizing it into an alpha helix or cyclizing it, we effectively shield all of its weak points from the hostile environment of our digestive enzymes. We are pre-organizing its shape so it binds flawlessly to its target while shrugging off rapid degradation. And we can't forget about PEGylation. By attaching specific protective polymers to the peptide, we basically throw an invisible shield around the drug. It significantly bumps up the effective molecular weight of the peptide. Why does that matter? Well, it essentially tricks your kidneys so they don't rapidly filter it out of your system. This chemical camouflage extends the drug's half-life so dramatically that patients might only need an injection once a week instead of every single day. That is a massive quality of life improvement. Finally, Section 5, the future of peptides. What is next for this booming $70 billion market? With all of this incredible biochemical hacking we've discussed, the horizon for therapeutic peptides is basically boundless right now. Armed with this newfound stability and pinpoint precision, the pharmaceutical industry is accelerating fast. There are over 170 modified peptides currently in active clinical development, and we're moving far, far beyond just treating diabetes or doing hormone replacement. Active research right now is tackling oncology to fight tumors, creating non-addictive treatments for severe chronic pain, developing life-saving interventions for sepsis, treating broad metabolic diseases, and even exploring cutting-edge antimicrobial applications designed to battle the looming global crisis of antibiotic resistance. It really leaves us with a truly thrilling prospect to ponder. As we continue to master this Goldilocks molecule, if we can literally rewrite the genetic code to build stabilized, armored peptides that nature never even intended to exist, what once incurable disease will we conquer next? The very building blocks of life have become the most powerful tools in our medical arsenal. Thank you so much for joining me for this explainer. I hope this helped you decode the incredible microscopic world of therapeutic peptides.